This is an optional video that goes over mistakes that can occur in meiosis, as well as a comparison of mitosis and meiosis. So I want to start with reviewing mistakes that can happen in meiosis. So if you look at the back of your big sheet, okay, what happens when meiosis goes wrong? As I said in the previous video, when meiosis goes wrong, we're talking about something called non-disjunction. Okay, so if you remember, during meiosis 1, something happens here. Okay, the thing that's supposed to happen here is crossing over between homologous chromosomes. That's supposed to happen during meiosis 1, right? But then what's not supposed to happen is that these chromosomes get stuck together. Okay, what's supposed to happen is what happens over here. Okay, we have separation in meiosis 1, the homologous chromosomes separate. So we go from 2N, or diploid, to N, or haploid, okay, by the end of meiosis 1. Sometimes, however, non-disjunction occurs, so the chromosomes get stuck together. If we follow the rest of meiosis through in non-disjunction, if chromosomes get stuck together, some sex cells will have extra chromosomes, because remember, gametes is a fancy word for sex cells. Some sex cells will have extra chromosomes, and some sex cells will be missing chromosomes. Right? This says extra, this says missing chromosomes. So if you have extra chromosomes from these sex cells, when the sperm and the egg unite and form an offspring, this offspring can have extra chromosomes. And this chart right here is called a karyotype. It is a chart that shows chromosome pairs. And you can see it shows all the homologous chromosome pairs, right? Here's your chromosome from one parent. Here's your chromosome from another parent. That's a homologous pair. Here's your chromosome from one parent. Here's your chromosome from another parent, and so on. So your entire karyotype shows the charts of chromosome pairs. You have 23 pairs of chromosomes. Karyotypes are useful for detecting genetic disorders because if you have an extra chromosome called a trisomy, you might have something like Down syndrome if it's chromosome 21. If you have an extra chromosome 18, that's called Edwards syndrome. If you're missing an extra Y chromosome, that's a monosomy. That is an example of Turner syndrome. Okay, so non-disjunction in meiosis, if the chromosomes don't separate correctly, you can have a genetic disorder occur. Other ways you can have genetic disorders occur is when you have chromosomal mutations. Okay, for example, here's the normal chromosome, here's the mutated chromosome. In a deletion, you're missing part of the chromosome. In a duplication, you have an extra piece of a chromosome. Now, it kind of makes sense. If you're missing part of a chromosome, then it makes sense that there would be problems, right? Because you're missing part of a chromosome, you're missing information. Why is it a problem if you have too much information, like in a duplication of a couple genes or in a trisomy? Well, too much genetic information can lead to excess protein buildup, which can cause problems, okay? You could have an inversion where part of the chromosome flips over and the genes are in the wrong order. This is a problem because we turn on and off DNA in different cells to specialize them, and if the DNA isn't where we think it's going to be, that could so if you're trying to make specific proteins and you can't find the DNA, that's a problem. Or the inversion could cut off in the middle of a gene, and that could ruin that gene. You can also have a translocation where part of one chromosome joins to part of a different chromosome. Okay, and this leads to problems because, again, the cell can't find the DNA where they think it's going to be. So these are problems in meiosis. Okay, that's one thing I got a lot of questions on. Another part of this optional video is a comparison of mitosis and meiosis because people were a little bit confused on this still. So in mitosis, we're making body cells. In meiosis, we're making gametes or sex cells. So we're talking sperm and eggs. So in mitosis, you start off with one cell, and you have one, well, you could have more than 4 chromosomes, but it'll just take me a long time to draw more. Like our body cells have 46 chromosomes. But let's say you have 4 chromosomes here. Okay? Let me pick a different color. Okay. So here are my 4 chromosomes right here. These chromosomes are homologous, meaning they have the same type of information. So maybe this one has gene A, big A, this one has gene little a, and so on. Maybe this one has genes B and C. Okay. So these 2 chromosomes are homologous and these 2 chromosomes are homologous. Okay, so here we're 2N or diploid. And chromosome number is 4 in this case. Well, we go through prophase, metaphase, anaphase, telophase, and we end up with 2 cells. And these 2 cells, the daughter cells, are identical to the parent cell, and they each have 1, 2, 3, 4, chromosomes. The difference is, so we're still 2N, we're diploid here. The difference is we started with 4 duplicated chromosomes, and we ended up with 4 unduplicated chromosomes. So the amount of DNA is different, but the genetic material is identical. 4 chromosomes, 4 chromosomes. Remember, I told you if you get stuck, you just need to count the centromeres. 1, 2, 3, 4. 4 chromosomes. 1, 2, 3, 4. 4 chromosomes. 1, 2, 3, 4. Okay? So that's mitosis. We're making body cells here. We're going to move into meiosis, but something they both have in common. They're both types of cell division, and they're both regulated by CDKs and cyclins for internal signals, and they have external signals as well. Okay, hormones and other ligands that control them. Now let's look at meiosis. Meiosis is going to look familiar to you at the beginning, right? Because it starts with a cell with 4 chromosomes, okay? And again, the same genes are present. But remember, we're going to have crossing over that's occurring here. So you can kind of see crossing over is occurring right here, okay? So in the beginning of meiosis, your cell is 2N or diploid. And the chromosome number is 4 duplicated chromosomes. After meiosis 1, you end up with 2 cells, and the homologous chromosomes have been separated. And the way they separate is based on how they line up in metaphase 1, right? Because if these 2 chromosomes were switched, the light green one would go with the blue. But because they're not, the dark blue chromosome is going to go with the, and remember, some crossing over occurred here. So we have a little a here, big A here, is going to go with the dark green chromosome. Now, crossing over would have occurred on the other chromosome pair too, but it's just would have taken me too long to draw. Okay, and here's my other cell that I've created at the end of meiosis 1. And again, here's one chromosome, and here is my other chromosome. Okay, so now at the end of meiosis 1, I produced 2 cells, and each of them have 2 duplicated chromosomes. So they went from 4 to 2. Okay, so we went from the full set of chromosomes to half the number of chromosomes. So we went from diploid to haploid. So both of these cells are in or haploid by the end of meiosis 1. In meiosis 2, it really just kind of looks like mitosis, really. So in meiosis 2, you can see the 2 sides of the sister chromatid split. So meiosis 1, the homologous chromosomes separate, bringing us from diploid to haploid. In meiosis 2, the 2 sides of the sister chromatid are going to split, creating 1, 2, 3, 4 haploid cells. And you can see they're all different in what genes they have because of crossing over. And crossing over would occur on all the chromosomes multiple times, so there'd be a lot of exchange here. So in the end of meiosis, you end up with 4 haploid or N cells, and in this case the chromosome number is 2 chromosomes. Now in a human that would be a little different, right? We talked about that in the video. In a human, in mitosis you would start with 1 cell with 46 chromosomes and you'd end up with 2 cells with 46 chromosomes. In meiosis you'd start with 1 cell with 46 chromosomes and you'd end up with 4 cells in the case of sperm with 23 chromosomes. In the case of an egg, you'd have 1 egg with 23 chromosomes and the other ones would be the polar bodies. Okay, so that is my review of mitosis and meiosis and mistakes that can happen during meiosis. Hopefully that helped answer some of your questions in the check-in. Thank you so much. Thanks for watching.